But if you look at the modern world around us, and everything humans have been able to build, from satellites to skyscrapers, there's no doubt we're more intelligent than monkeys. So what changed? What made our brains so different from those of other primates?
Hi, Neil Schumann.
Hi, Neil. It's Tom Verbacher.
Good to see
Welcome
you.
to the Infant Primate Lab.
So this is the monkey place,
This
huh?
is the monkey place.
Tom Verbacher studies monkeys in order to answer questions about our own brains. He's going to show me an experiment that demonstrates an important difference between our brains and the brains of a monkey. But the differences aren't what you might expect, especially when we're young.
Monkey on the set.
Monkey on the set. Neil, this is C.I. She's a 3-month-old pigtailed macaque.
This experiment tests something called object permanence. In other words, whether this monkey knows an object still exists when she can't see it. It's a developmental milestone for both monkeys and humans.
You're going to put a piece of fruit on the toy itself. You'll place it in the well,
She's looking at it. There.
and you'll move that to completely hide that toy.
Okay. Oh,
There you go.
wow.
There you go.
Wow. That is impressive. Just
Object
boom,
permanence.
boom.
3 months old, and already CI understands object permanence.
CI, you are good. Hi, Geneva. How are you? You are a cutie.
Geneva is the same age as the baby monkey. I'm going to do the exact same test on her under the expert eye of child development specialist, Professor Susan Speaker.
She needs to reach for him, show that she's interested.
Oh, I think we got
She's
interest.
got a good reach.
You do. Look at you. Oh, Geneva, you like those. Hello.
Now, Neil, can you get them away?
Oh, I'm an expert at this. Can I take that for a second? Sorry, be temporary. Yeah, you are so good that way. I'll pop it in. And
Make sure she's watching.
she's watching. Now we'll cover it up. Watch this.
And she wonders, what did you do, Neil?
What toys?
I don't know where they went.
Wow, that is amazing.
Where did they go?
It is incredible. If it's not there, it doesn't exist.
Right.
That is amazing.
So how come a baby monkey beats a baby human in this test?
These tests are showing us how rapidly a particular region of our brain is wiring up in development. And it's showing us comparatively that we wire up much slower in this part of the brain than do monkeys.
Compared to other primates, we humans have an unusually long childhood, even if you take the different lifespans into account. This should be an evolutionary disadvantage. But in fact, it's one of our greatest strengths. During childhood, our brains are being shaped by our environment and by our experience. Extending this phase gives us longer to learn and pick up new skills. Some think that this is a key part of what makes us so smart. But there are other factors too. The brain is made up of vast networks of nerve cells called neurons that carry, process, and store information. The part of the human brain responsible for higher thought, the cortex, contains more neurons than that of any other animal on the planet, an astonishing 16 billion. These neurons are organized in a uniquely powerful way. They're unusually interconnected. This means we can do things that other primates can't: build complex tools, compose symphonies, and even investigate how our own brains work. But before we get too carried away with our own cleverness, I want to inject a bit of humility. That means leaving monkeys behind and going back to where the series began, my anatomy lab in Chicago, and my favorite subject, fish. The brain of a fish and the brain of a human have more in common than you might think. This is a trip to the beginning. This is as basic as it gets. We're going human fish, represented by a cartilaginous fish, a shark on the right. And what you have is, you see the muscles and nerves and organs inside the shark head. But importantly, you see this yellow tissue. This is the front end of the spinal cord. It's the brain. Doesn't look like much, but it's the shark brain. This is the human brain. This is where it all happens for us, where our memories reside, where our fears, our loves, our hates. It's all in here. The human brain, really at the surface level, looks nothing like what's sitting inside the shark head. Yet when you know how to look, you find a very profound similarity. And that similarity lies in the overall architecture of the brain itself. In the human, we have a forebrain. And that forebrain consists of this folded tissue here, where much of our conscious thought happens. And then there's a midbrain, this walnut-sized area in the middle. And then a hindbrain. That's a fundamental division. We see that in development, and we see that based on where the nerves exit the brain. Well, if you follow the similar nerves in a shark, what you see is sharks too have that same structure. A forebrain, a midbrain, and a hindbrain. There's a fundamental architectural similarity among all brains. The brains of people, the brains of dogs, the brains of cats, the brains of monkeys, lizards, frogs, salamanders, trout, bass, even sharks and fish are similar. Each one of them has a forebrain and a midbrain and a hindbrain, despite the fact that those brains are often very different in function and form. But that's not the end of the story. The fundamental architecture of our own brains can be traced back even further than fish, to a surprisingly primitive animal that doesn't even really have a brain. But I've got to find these tiny creatures first.
Right, okay. Well, we could start trying around
X
here.
marks the spot.
X marks the spot. Let's give it a go.
This is Peter Holland, a world-renowned geneticist and head of zoology at Oxford University in England.
Okay, so what do I do?
Just shake it.
Shake and bake, huh?
I'll make it look so easy.
That? No?
No. They'll be pretty obvious because they'll really swim around. The water makes the digging slightly harder.
It does.
You got one. There's an amphioxus.
We got any? Whoa, look at that little guy go. Hey!
Just dip it in the water again, you'll see it'll flick around.
Well, they really flick.
There you see. There he is.
Hey, it's beautiful. Look at that. Which is the front?
It doesn't look like much, but this tiny creature, called amphioxus, has much to tell us about our own brains.
That is cool. Okay, that ranks as cool.
Okay, so let's take a look at a couple of these.
So I brought this from the anatomy lab in Chicago. This is a human brain. When I look at this, I don't find any obvious similarities to that.
Deep in the genes of this animal and the development of this animal, and deep in the genes of us and the development of us, there are the similarities.
They're pretty active. They're pretty active.
Holy cow. Oh, that is beautiful. God, they're so clear too. You can see right through them. You know, it's amazing.
Amphioxus lives in the sand of the ocean floor, filtering algae out of the water. It's from an incredibly ancient group of animals. We found fossils that look a lot like this in rocks that are over 500 million years old. They are a window into what came before the first fish. And crucially, they don't have anything like what we'd call a brain. The simple nerve cord that runs the length of their body ends with just a tiny swelling invisible to the naked eye.
If you look at the front end of this animal, you don't see it all expanded into a large skull or brain region. It's just pointed.
But if you look at the genetic makeup of these ancient creatures, you find something truly remarkable. To make the forebrain, midbrain, and hindbrain that we saw in our lab, you need a series of control genes to tell the cells within the developing brain where they are. They're like a zip code. Those same control genes that make our own brains are active in amphioxus. But here, they're simply building the primitive nerve cord and the tiny swelling at its front. This means that the genetic roots of our complex primate brains existed in much simpler creatures that first arose over 500 million years ago.
I think it gives us a glimpse into where our brains came from, into the basic organization of the brain of our ancestors.
I mean, I find that mind-blowing.
And he's just an exciting animal.
He knows you're talking about him. See, he's going.
That's right. He's going. You can see those muscle blocks really working there. You know, and I think to an evolutionary, but well, he's jumped off, guys. Sorry.
He's going Tiktaalik on us.
Sorry.
He's going land living. He's going rogue.
Yep, he's gone for the next transition.
Exactly. He's in the Devonian now.
The search for our inner fish has taken me from high in the Arctic.
Here was the snout of exactly the creature we were looking for.
To the plains of Africa.
Yeah, I just want to run down here and start collecting fossils. Let's go.
As well as deep inside our own DNA.
And today we saw a little tiny worm living in the mud here in the bay that contains genes that we have that sculpt our own brains. The organ that gives us many of our unique properties.
We've seen how our distant relatives, from worms to fish, from reptiles to early primates, have defined our bodies. At each new stage of our journey along the tree of life, our ancient animal ancestors have reconfigured what's gone before. And that's how we ended up as the intelligent creatures with all our quirky flaws that dominate the planet today.
There's something incredibly profound, and I think beautiful, in the idea that inside every organ, cell, and gene of our body lie deep connections to the rest of life on our planet.